Okay, good morning everyone. I'm just going to put the timer on because I don't want to go over time. Hope you're all doing well today, enjoying your breakfast. I'm delighted to present to some of our findings from my 2021 Raine priming grant. So that was funding that was provided to our group between 2022 to 2024. So how time flies, but we have received some really good outcomes from the project, and I'll be sharing that to you in the 5 minutes I've been allocated. So just I thought I'd give you a brief introduction about myself. So I was a Raine primary grant recipient in 2021. Currently I'm a research fellow at Curtin University and the Parent Institute. So I'm very privileged to be working in translational neuroscience, where essentially we're trying to bridge findings from our biodiscovery lab findings to clinical translation. And that's in order to reduce disease burden in a number of neurodegenerative conditions. So as you can see just in here on the left, Alzheimer's disease, multiple sclerosis, migraine, Parkinson's, and a number of other disorders as well. So I'm currently co-leading a large group of preclinical and clinical scientists, but we're also very privileged to be working with a number of clinicians, which makes a lot of this research possible. We also collaborate with a number of national, international partners, and it's because of this collective expertise that makes our research possible. So MS, what is MS? I'm sure a lot of you will know that MS is a progressive neurological condition that basically affects myelin, which is a protective fatty coating that coats axons and neurons. Now, over 33,000 Australians are currently affected by MS, and nearly 3 million individuals are affected by MS globally. Now, as you can see in this figure here on the right-hand side, it's that breakdown of this myelin, which is primarily made up of lipids or fats, which gets broken down over time. And this coincides with disability, worsening of symptoms, which includes motor impairment, fatigue, loss of cognitive functioning, and a myriad of other symptomology. Now, whilst there are a number of disease-modifying therapies that are really potent at reducing disease burden through immunomodulation, what is still missing critically is that there's no cure to MS, which is really based on trying to find any kind of treatments, interventions that can promote this remyelination to help prevent demyelination from occurring, so the loss of this fatty coating, but as well as helping promote this repair process. So that's what our group has been working on for the last 5 years. So essentially we asked a very simple but really important question. What if we can provide these lipids through diet or through intervention, and provide these lipids that can get into the central nervous system and help promote the remyelination process? So through the support through the Raine Foundation and other funding partners, what we were able to do was we tested out a few different lipid-based interventions. So we collaborated with the group, with the Baker Institute over in Melbourne. We tested out a couple of formulated lipid interventions in mouse models of MS. This was basically to test if prophylactic provision of these lipids was able to help remyelinate and improve motor function in these different animal models of MS. What we also were able to do was we characterised the plasma lipidome in a longitudinal cohort of patients from MS, and that's through the Parent Institute. This is patients with different phenotypes of MS that were followed for up to 10 years, where we're able to test out the plasma lipid species along with disability progression. So what we found from this work was really exciting. We found that one of the specific lipid formulations in this mouse model of MS was able to improve motor function, it was able to reduce demyelination and improve remyelination at the same time. So this set up a really strong clinical foundation for us to take up these studies in a clinical trial. So I'm pleased to share with you that we've just got recent ethics approval to run the Repair MS clinical trial, and that's to test out this specific lipid compound in people with early MS. Now, ideally, we'd like to test out this efficacy of this particular compound in people with different subtypes of MS, but people with early MS have the greatest capacity to remyelinate. So it was an obvious choice that we target people with early MS first. So we're hoping to launch this trial in early next year. So if you do know of any people or any networks who would be interested in participating in the trial or learning a bit more about the trial, please get in touch with me. So I'd just like to say my thanks to the Raine Medical Research Foundation for supporting early career researchers like myself to build our independence, leadership skills, as well as continuing a research program. Also, I'd like to thank my research team. A lot of this work obviously wouldn't be possible without their help, as well as our collaborators and our funding partners, which are indicated on the left. And lastly, I'd just like to congratulate all of the recipients that will be announced as well later on this morning, and good luck with your projects. I look forward to hearing about the findings later. Thank you so much.